I'd like to thank you all for coming today. It's a big day. Vadim is going to defend his dissertation, and now the important guys are here, so
now.
Get started. Vadim I was thinking, okay, he hasn't been here as long as I have, but he's been here for a while. So he got his degree in 2000 from UCI Biological Sciences, and then stayed locally, worked at Neo Therapeutics for a couple years, doing behavioral work in rats, and then joined Hans Kirsten's lab, and also did some more behavioral studies with Hans before he started graduate school in 2003. And upon entering graduate school, he went through a few laboratories. In fact, really sort of had to wander around a bit before he found us. So he didn't join the Lynch and Gall lab, but Gary Lynch is acting as his immediate supervisor in the studies. But he didn't join us until 2005, the fall of 2005. So it's been a very intense 2.5 years where he has done a lot of behavioral work and microscopy in particular. He joined the lab right when we were engaged in studies where we were looking at reorganization of the actin and spine actin cytoskeleton in association with LTP, and really wanted to see if the same things happened during learning. And so the task fell to Vadim and some other trusty collaborators in the lab to develop behavioral paradigms that we could use to study the effects of learning on spine neurochemistry, in particular spine cytoskeletal reorganization. And as you'll hear, he succeeded in establishing a behavioral paradigm in the laboratory. Worked for a year on a project that didn't come to fruition, so you're not seeing everything he did. But we totally got our stride and figured out how we were going to look at the spine actin reorganization. So, Vadim, I don't know if you ultimately changed your title. The title that I have is he's going to talk on learning activates actin regulatory pathways within the subpopulation of generally spine. Is that what we're using? Oh no, we changed it. The last I saw we had 3 options. He was having trouble deciding. So he's going to tell you about the studies where unsupervised learning induces regional synaptic changes associated with encoding of memory. And I'm very proud to introduce Vadim Dissertation.
Thank you very much for the introduction, and thank you all for coming here today to hear the talk. As Dr. Gum mentioned, we're going to be talking about unsupervised learning, which you guys see here in this picture of rat undergoing unsupervised learning. And this induces regional specific synaptic changes associated with encoding of memory. But let's talk about learning and memory for just a second. Learning is a continuous process that we encounter on an everyday basis. It involves acquisition of a great deal of information about our environment. The memory for such information requires high capacity storage mechanism. Currently, LTP is a widely accepted model of such high capacity storage mechanism because it produces immediate and prolonged increase in synaptic strength and structure and satisfies the high capacity requirement because it is synapse specific. The study of this high capacity learning was once a major goal in psychology. However, as Hebb eloquently put it, the task of understanding behavior and reducing its complexity of thought to a mechanical process of cause and effect is more difficult than that of any other scientist. Thus, in recent times, neurobiologists oversimplify behavior in order to correlate it to neural processes. Low-capacity learning paradigms were set up, which involve specialized, stressful, or unusual circumstances, such as shocking the rats or having them swim through water, which is analogous perhaps of sticking a fork in an outlet or being dropped off in the ocean and trying to find safety. So it doesn't really relate to the kind of learning that we do on an everyday basis, which involves continuous flow of information such as we're exposed to at this moment. It involves everyday interactions that are usually found in nature. Sometimes it involves navigation through space and learning the spatial relationship between different cues and their importance. It results in numerous complex responses and change in response as the environment changes. However, recent technological advances have allowed us to study this high-capacity learning that is found in nature by the ability to collect and analyze complex patterns of everyday learning behavior. Thus, we use these advances in technical advances in order to develop a novel behavioral task that studies this high-capacity, everyday naturalistic learning. we've established measures of learning and memory in this behavioral task, and we're able to use this task to characterize the disruption of learning that occurs in models of human cognitive dysfunction. Then we're using the behavioral task, we made a leap from behavior to neurochemistry, and in order to determine high capacity learning results in LTP-like changes that occur within individual synapses. These studies opened the way of creating maps of where exactly in the brain the synaptic modifications occur during learning. This is an example. This is a picture of unsupervised learning paradigm. It consists of an open field, as you can see here, a dark enclosed compartment where the animal can find its refuge, and two objects in the open field. Young male adult rats are exposed to 4 days of learning for 60 minutes each day in this paradigm. On day 1, they're exposed to open field only to familiarize themselves with the room and the open field compartment. On day 2, however, the complexity of the environment is introduced with the dark compartment and objects being added. On day 3, it remains as the same as day 2, and on day 4, we substitute one of the objects for a novel object. We measure several coordinates of learning and memory, such as percent time moving, number duration of individual moves, distribution of activity for different compartments over time, and the duration of time the animals come to investigate the objects. This technological advance I was talking to you about is a software by Vision XD, which is able to track animal movements in real time and give us the data. As you can see from examples of traces from 4 animals which are ran simultaneously, the rats, each rat performs different pattern of activity in the boxes. As you can see, from 2-minute traces, 10-minute traces, and 60-minute traces. What is seen from these traces is that animals do 2 things. They're either moving, as shown in gray, or pausing, shown in orange circles. And you can see that they pause mostly in these regions of interest that they explore, such as the dark compartment where they can overlook the open field, the objects, and the corners. Thus, as you can imagine, analysis of such complicated trajectories was not an easy task, but we found some measures of learning that can show day-to-day changes.
First, could I just ask a question about?
Yeah.
Is this software tracking like a point on the mouse's nose, or is it tracking the whole mouse?
On the middle of its body. There's options to also track its nose, but we didn't have this option. We only had an option where it tracks the middle, its midpoint. It calculates where, the midpoint of the animal's body.
Okay. So when it's associated with one of the objects, you can't really tell whether it's sniffing or farting. Like
exactly. That's why we set up these regions, right? And we calculated the time the animals is pausing in this region versus the time they're actually moving through it. Thus, we can tell when the animals is actually investigating versus just passing through this region.
And the tracking works in the dark too?
In the dark too, because overhead we set up the CCD cameras, which have very low threshold seen in the dark, and we have infrared lights illuminating the open field, which provides illumination into the dark compartment so the camera can see through it. So the dark compartment is very dark and closed, and animals can't see through it, but the camera is scanning because we have infrared lights illuminating the field.
So why is this better than the usual, like a little bulb on the rat's head and LCD light being tracked?
Well, it's because this software can collect the data in real time, and you can output a lot of statistical measures right immediately following the test.
But you can put a little LCD on the rat's head, and you can do the same kind of tracking, right? So just wondering, what's the advantage?
Well, advantage is you don't have to put anything on the rat's head, so they
It's fair enough. But on the other hand, you lose specificity, as you just said.
Exactly, yeah. So in the future, we're definitely looking to improve this by adding the nose module, because this software can actually tell the nose and sniffing behavior. But this is something that where we've learned after doing these tests that we can improve, something that we can improve upon while gathering this data. So looking at the percent time moving from the rats from the beginning of the test, we can see the rats start out with very high exploration levels of the environment when the novelty is introduced on day 2. This movement decreases over time as the rats become familiar with the environment. It reflects the reduction in the duration and frequency that the rats make, of moves that the rats make, and a shift of their behavior into the dark compartment over time on day 2. Looking at the traces on day 3, we can see that the animals start out with much less percent time moving on day 3, showing that the animals show experience-dependent changes from day to day in their behavior, suggesting that exploration on day 2 produced some memory of the environment where they didn't feel like they had to explore it to the same degree at the beginning of the test on day 3. To further characterize the animal's distribution of time during their explorations on day 2 to day 3, we quantified the amount of time that they spend in each compartment. Whereas obvious that animals spend a great deal of time in refuge and in the open field, perhaps moving to the compartments where they want to investigate. Looking at a difference in distribution of behavior from day 2 to day 3, we do not see any evidence of changes as a result of experience from day 2 to day 3. They spend the same amount of time in each compartment on each day, even though they're moving less on day 3, suggesting that the young rats develop a stereotype strategy for exploration. We wanted to further characterize and determine if this exploration produced enough knowledge of the environment in order to notice when novelty is introduced. So we introduced a novel object on day 4 and measured the animal's contact time with this novel object. So what we find is animals respond significantly to novelty by increasing the contact time with the object on day 4 compared to the time they spend with it on day 3. This is also very different from the time they spend with a familiar object. We established a certain criteria of novelty detection in order to figure out what percentage of these rats notice novelty in the environment. This criteria says that if an animal spends 30 seconds or more, 30 seconds or greater time on day 4 as on day 3, and greater than 30 seconds time as they spend with a familiar object, they become a detector, thus they detect the novelty. So applying these novelty detection criteria, we find that 83% of detector rats satisfy this criteria for novelty detection. Thus, the majority of the rats have increased their time significantly exploring this object. So these studies have shown that animals learn in their environment during the exploration time. Next, we wanted to find out if these measures of learning and memory are dependent on the NMDA receptors and LTP-like